Hello, good morning students. Today we'll study cleavage. Cleavage is fast division of zygote to form the blastula. So cleavage refers to early mitotic division that divides up large volume egg cytoplasm. The early zygote is unique in its so large shape. Most cells undergo a period of growth between cycles of mitosis, but that is not true for early cleavage stage blastomere. With each division, the cells get smaller. So in this graph, we can see that during the cleavage, the number of cells, they increases abruptly to large number, and later on, this growth, it decreases. The first cleavage of frog egg was observed by Swammerdam in 1738, whereas the entire process of cleavage in frog's egg was studied by Prevost and Dumas in 1824. With the development of microscopes, cleavages and further stages were observed in the eggs of sea urchins, starfishes, amphioxus, as well as the hen's egg. The pattern of embryonic cleavage is determined by number 1, position of the mitotic spindle, second, amount and distribution of yolk. So these are the two important features which determines the pattern of cleavage. The yolk tends to inhibit the cleavage, it slows it down or actually prevents complete cleavage. Yolk is an adaptation of those animals that go through more or less of embryogenesis isolated from any food supply. Some animals like sea urchins have relatively little yolk because they rapidly develop into free swimming larvae that acquires nutrients from the environment. Other animals such as marsupials are born prematurely but are provided nourishment in the parental pouch, whereas the placental mammals, they develop a specialized organ through which the embryo is nourished throughout development and so after have a little yolk. Now types of eggs based on the yolk. Isolecithal, sparse, very less amount of yolk, evenly distributed yolk, for example sea urchin and mouse. Second is mesolecithal, moderate amount of yolk, often unequally distributed, for example frog. Third, telolecithal eggs, dense yolk concentrated at one end, for example reptiles and birds. Then centrolecithal. Centrolecithal is yolk is concentrated in the middle of the egg, for example insects. Many eggs are polarized with a yolk-rich pole, termed as vegetal pole, and yolk-poor pole known as animal pole, for example frog. The zygotic nucleus is generally displaced towards the animal pole. Cleavage. Zygotes with relatively little yolk, that is isolecithal or mesolecithal, cleave holoblastically. The cleavage furrow extend all way through the eggs. Telolecithal and centrolecithal egg undergo meroblastic cleavage, where the cleavage plane extends only to the accumulated yolk. In centrolecithal eggs, cleavage is meroblastic and superficial, whereas in telolecithal eggs, cleavage is discoidal. Now there are several types of cleavage symmetry. Number 1, radial, seen in the case of echinoderms and amphibians. Spiral, mollusca and annelida. Bilateral, acidaceans and tunicates. Rotational, mammals. Now see, this is how the cleavage pattern is there. Number 1 is holoblastic. Holoblastic cleavage is shown by the isolecithal eggs or the mesolecithal eggs. In isolecithal eggs, as I've told you, there are 4 types of cleavages. Number 1 is radial. In radial cleavage, the zygote it divides into 2 and then these all are the cleavage which are the radial cleavage. Then is the spiral. Spiral means after every cleavage it rotates, and the spiral symmetry it is maintained. Bilateral cleavage. The cleavage is bilateral when we cut the any stage into two equal halves, right? The cells, they divide, the blastomeres, they divide, and they divide in, you can say, left and right side, two sides, bilateral. Then is rotational. In rotational cleavage, after every cleavage, rotation of the blastomere is there, and as a result of which the blastodisc is formed in the case of the mammals. Now coming to the mesolecithal cleavage, it is also radial. It is present in the case of amphibians. In the case of meroblasticial cleavage, the yolk is present at the vegetal pole, and as a result of which, when the cleavage it starts from the animal pole, it does not complete before the second cleavage it initiates, as a result of which the upper cells, they are the smaller cells, and lower cells, they are the larger cells. Now coming to the meroblastic cleavage is shown by telolecithal or the centrolecithal eggs. In the case of telolecithal, we can see the bilateral. Bilateral means a symmetry it is maintained, left and right similar type of division it is there. And in the case of fishes, reptiles and birds, we can see the discoidal type, that is only in the disc the blastomere they are formed, and in the lower portion, because yolk is present, no division is there, so embryo is formed at the disc portion only. And centrolecithal egg, which is shown by the insect, it is superficial. Here what happens, first of all the nuclei, they divide into large number, and then later on, after 14th stage, after the 14th division, the division of the cytoplasm is there, that is the cytokinesis is there at the margin. So this is regarding in the case of the centrolecithal eggs. Now one by one we'll explain all the three. Number one is holoblastic radial cleavage. In this case, complete egg, complete division. See, this is animal pole, this is vegetal pole, right? So first cleavage meridional, नीचे तक. Second cleavage again meridional. ठीक है, जैसे एप्पल लिया, काटा 1, काटा 2, ठीक है, तो बराबर हमने 4 हिस्से उसके कर लिए लॉन्गिट्यूडनली, राइट? एंड देन हमने बीच में से काट दिया, इक्वेटोरियली काट दिया, तो 8 हिस्से हो गए. So in this way the cleavage it proceeds. Holoblastic, पूरा का पूरा जो the one you can say the zygote है, पूरा का पूरा जो है वो cut down होगा. That is the radial cleavage, right? Then coming to the next is holoblastic mesolecithal radial cleavage. That is in the case of amphibians. See here, the first cleavage is there. Before the finish of first cleavage, the second cleavage it initiates, right? So before the completion of second cleavage, the third cleavage it initiates. When the third cleavage initiates, it forms upper micromeres, lower macromeres, right? So normally what we can say that the speed of cleavage is variable. So in the animal pole, the cleavage proceeds at 1 mm per minute, while in the vegetal pole, because yolk is present, it proceeds 50 to 100 times slower. So as a result of which it takes time to complete its cleavage. So say here the cells they are larger, here the cells they are smaller. So in this way the holoblastic radial cleavage is there in the mesolecithal eggs. Now coming to the meroblastic cleavage, in first of all the discoidal cleavage. So you see here, this is the zygote. So in the upper disc portion only the cleavage of cells they are there. So large number of cells they are formed over the upper portion only. So telolecithal eggs, they results in the meroblastic discoidal cleavage. So it is restricted to blastodisc at the animal pole of the egg only. At early cleavages, because cleavage cannot proceed through the yolk, the blastomeres are continuous at their vegetal margins. Now coming to the centrolecithal cleavage is different in the case of insects. Here yolk is present in the center. So in this case what happened, the zygotic nuclei divide without cleavage, right? So nucleus undergo karyokinesis, that is the mitotic division of the nucleus. So large number of naked nuclei they are present. They are known as energids. So large number of them they are present, right? Then what happens at this stage, we call it as syncytial blastoderm because the nuclei they are present in the same cytoplasm. Later on, after 14th nuclear division, the cellularization it occurs and it leads to the formation of cellular blastomere, means cytokinesis is there. That is the cytoplasm, it divides and it forms the cellular blastomere in the periphery. So this is there in the case of the insects. Now, significance of the cleavage. Cleavage, as you know, it is the repeated mitotic division of zygote to form a solid ball of cell known as morula, which later on changes into hollow ball of cell known as blastula. It converts a unicellular zygote into a multicellular embryo. So cleavage is very important. It maintains the cell size and nucleocytoplasmic ratio of a species because the cell size of the egg is very large, but the size of the individual is very small. Cell size of individual is very small. So it maintains nucleocytoplasmic ratio. So cleavage produces large number of cells or blastomeres required for building of the offspring's body. During cleavage, quick mitotic division of blastomere occurs, following which there is no growth of blastomeres. The cleavage brings out the distribution of cytoplasm among the blastomeres. So these are the significance. Now coming to the mechanism of cleavage. So in the cleavage, very important thing is when we look at the cell cycle or the mitosis, what we say that there is M phase, followed by there is G1, then S phase, then G2. In the G1 phase, the synthesis phase is there. In the S phase, the division of the nuclear material is there, whereas in the G2 phase, again the synthesis of the material for the organelles is there, and then the division of the cell is there. But during the cleavage, what happens, the G1 and G2 stages they are absent. The M stage directly follows the S stage and the S stage directly follows the M stage, as a result of which no growth phases there. The size of the cell it becomes smaller and smaller after every cleavage till the nucleocytoplasmic ratio is maintained. This is the mechanism. Thank you www.google.com for providing us beautiful pics so that we can explain the topic well. Thank you.